going to talk about ADP ribosylation of proteins and its implication in health and in the disease. So Michael, I stop sharing my presentation, and then you should be able to share your own screen.
Thanks a lot, Tiziana. Is my screen visible?
Yes, it is.
Good. So good afternoon, everybody, and thanks to the organizers for giving me the opportunity to speak here this afternoon as the first speaker. As Tiziana said, I will be talking a little bit about ADP ribosylation and characterizing this modification using mass spectrometry. As the first speaker, I also thought that something is, sorry, wrong presentation.
Too many presentations.
Yes, exactly. Here we are. So as a introduction briefly with the PTMs, so protein modifications are, as many of you know, very important regulators in essentially any biological process, and they're very dynamic and therefore also quite challenging to study. And of course, mass spectrometry is very well suited for studying most of the. Yes.
I'm not sure if we see your right presentation already. We still see the slide that starts with AF1521 based proteomics. This is better.
But now you're muted, probably.
Yes. Sorry about that. I think we are ready to continue. So yes, protein modifications, as I said, are very dynamic regulators of any biological process. And the modification I would like to talk about today is a modification that is called ADP ribosylation that my group has been focused on for the last couple of years. It's a modification that is catalyzed by a family of enzymes known as PARPs. And in human cells, we have essentially two kind of families that can catalyze these modifications. We have an intracellular family consisting of 17 members, and then also a plasma membrane embedded extracellular membrane family consisting of essentially 4 members, but of which 2 are only active. And what they do is essentially they use NAD as a substrate and then transfer ADP ribose units onto protein targets. But for many of these PARP enzymes, it's not really fully known what the biological function actually is. So if you go into the literature, you will see that many of these proteins are reported to be actually active in many different processes, and it's not really clear essentially what the individual function actually are. And likewise, many of, if you go into the literature, the actual amino acids that can be modified with ADP ribosylation is also kind of diverse. So in the literature, you will find information about up to 11 amino acids that can be targets of this ADP ribosylation modification. So we thought there would be a need to kind of, develop an unbiased proteomics strategy for studying ADP ribosylation in order to try to infer new insights into ADP ribosylation. And to this, we essentially started looking at the most widely known PARP modification, which is PARP1. Is essentially the one only enzyme that is quite well known, and its molecular function is quite well described. So it's a very important component of the DNA damage response, where it plays very specific and important roles in repair systems like the base excision repair. So in here, upon DNA damage, PARP will actually recognize a damage to DNA and will bind to it, and upon this binding, PARP1 will become activated and start catalyzing ADP ribosylation both on itself but also on other factors. And due to this importance in the DNA damage response, specific inhibitors, the so-called PARP inhibitors, has been developed that is now widely used in the clinic for treatments of various cancers. And the philosophy here is that if you inhibit PARP, you essentially inhibit the certain repair pathways, or particularly the base excision repair, and prevents cells from repairing single-strand breaks. In normal cells, there's other mechanisms that can take over and continue the repair, but in certain cancer types, where homologous recombination is also impaired, PARP inhibitors can be used as a very specific treatment to kill certain types of cancer cells. And if we look at the catalytic preferences for PARP1, it's been historically known or described that the PARP1 very widely catalyzed ADP ubosylation on glutamic and aspartic acids. But the function of PARP1 and essentially what proteins are modified with ADP ubosylation has been not so well described. So therefore, we developed an MS approach to kind of study ADP ubosylation, and for this we use a macrodomain that is called AF1521. So the macrodomain can specifically recognize the terminal moiety of this ADP ribose unit. So the macrodomain can essentially be used to enrich both mono ADP ubosylation, but also ADP ubosylation occurring in poly chains. And the strategy that we developed follows very basic principles similar to many other bottom-up approaches for studying PTM. So essentially, we digest the proteins into peptides. We then use enzyme called PRG that can reduce poly ADP ubosylation chains into mono ADP ubosylated counterparts, such that we essentially have mono ADP ubosylated peptides. And then we can use this AF1521 enrichment strategy to fish out the mono ADP ubosylated peptides, and then we can analyze these by mass spectrometry. Now, the mass spec challenges associated with ADP ubosylation is that it's a very labile modification, far more labile than, for example, phosphorylation. So we need electron-based fragmentation methods in order to actually pinpoint the localization of ADP ubosylation. And we demonstrated this a couple of years ago, where we actually compared the localization power, so to speak, of ETD compared to HCD. And what we essentially could see with HCD, that HCD is not at all able to localize ADP ubosylation on these enriched peptides, whereas ETD works really well and generates very nice ETD spectra, from where we then can deduce exactly which amino acid is modified with the ADP ubosylation. And what came out of our study here, global study, was essentially that what we found was almost exclusively ADP ubosylation localized to serine residues, which of course raised questions regarding what has previously been reported for PARP1. And what we could see is that these serine ADP ubosylation events are very specifically catalyzed upon damage treatment with, for example, oxidative damage. And when we treat with a PARP inhibitor, the catalysis of serine ADP is reduced again. So very strongly pinpointing that this serine ADP ubosylation is catalyzed by PARP1, that is the main target of these PARP inhibitors. Similar picture can be depicted here in a heat map. So essentially, untreated cells do not really show any ADP ubosylation. When we induce with H2O2, we see massive induction of a serine ADP ubosylation, and when we treat with PARP inhibitors, all this is again abrogated. And what we then essentially found was more than 1,000 proteins that was actually modified with this serine ADP ubosylation downstream of PARP1. Of course, hereby expanded quite significantly on the knowledge of ADP ubosylation that was available at that time. And what we also could see was that the serine ADP ubosylation very strongly localized to a KS motif. So serines really preferred having a lysine in front of the serine residues. We also saw indications that there could be a diglycine motif, but when we looked a little bit further into this, we could see that this was actually an abundance bias. So what we found was a lot of RNA binding proteins being targeted with ADP ubosylation, and those RNA binding proteins are very rife in diglycine motifs, and essentially they're also very abundant. So it was more of an abundance enrichment rather than specifically preferences of the PARP enzyme. And we could also see that very reassuringly that all the majority of the targets we identified indeed are nuclear targets, which aligns very well with the known nuclear localization of PARP1. And of course, we could predict, or we could assume some structural preferences of the modifications as well. So why is it that we see essentially serine ADP ubosylation, where the literature and, for many years people have reported glutamic and aspartic acids to be modified? Well, there are probably several reasons for that, but seminal work from the lab of Ivan Arhel described that PARP1 needs a specific cofactor in order to catalyze its ADP ribosylation activity, a cofactor that they called HPF1. So the hypothesis is that without this HPF1 factor, PARP1 prefers to catalyze ADP ribosylation on aspartic and glutamic acids, and when HPF1 is present, it prefers serine ADP ribosylation. Now, for many decades, most of the work related to PARP1 was done using in vitro reactions, and of course, these reactions were done without the presence of this cofactor because it was only identified recently, which could kind of explain why many reported glutamic and aspartic acids as serine or ADP ribosylation targets. To explore this a little bit in more detail, we also performed similar in vitro reaction experiments now coupled with mass spec analysis. So we took PARP1, recombinant PARP1, and incubated it in the presence and absence of HPF1 and also in the presence and absence of a known PARP1 substrate, histone H1, and then analyzed this using ETD. And essentially what we saw was that when HPF1 is present, we see massive induction of serine ADP ribosylation, both on PARP1 itself, but also on H1, the known substrate of PARP1. Whereas when HPF1 is not present, well, we do see a little bit of signal referring to glutamic acid ADP ribosylation, but it's essentially an automodification, a very low abundant automodification of PARP1 itself that we can see here. And of course, being in vitro reactions, it could be kind of an off-target effect, but nonetheless, we see at least a massive difference in overall abundance between the induced glutamic acids, glutamic ADP ribosylation in the absence of HPF1 and serine ADP ribosylation in the presence of HPF1. But of course, to explore this in a little bit more detail, we went into cells, and then we generated essentially knockout cells of HPF1 and also an enzyme called AIH3. So AIH3 was also identified by the Arhel lab to be the enzyme that can actually remove serine ADP ribosylations from target proteins. So to explore these two, the regulatory aspects of these two enzymes, we generated knockout cells, pulled them through our enrichment analysis, and then analyzed everything again using our mass spec approach. And what you can see here with the Western blot is that kind of reassuringly what we see when you knock out AIH3, of course, you see a massive endogenous increase in serine ADP ribosylation, very similar to the one you see when you induce just regular control cells with the H2O2. And this is also what we could see from the number of identified ADP ribosylation sites and also from the overall ADPR intensities. And again, what we could see is with the HPF1 knockout cells, we hardly see any sites and hardly any intensities, and that is essentially less abundant than compared to untreated wild-type cells. And when we did a PCA plot, which also nicely indicates this, we have the control cells here, and actually both the HPF1 knockout conditions, both the untreated and the treated ones, cluster very closely and very nicely with the control untreated. Whereas if you imagine the control treated with oxidative damage, they are of course massively induced with the ADP ribosylation, but not to the same extent that we see when we actually knock out the removing enzyme that is called AIH3. So essentially what we can see is that the control cells upon damage are actually moving towards, of course, the AIH3 knockout cells, which collectively tells us when we also look at the overlap between sites that HPF1 and AIH3 indeed are the two enzymes that regulate a very homogeneous serine ADP ribosylome, and they do it on a global scale. So you have PARP1 that can catalyze serine ADP ribosylation, you have in the presence of HPF1, and you have AIH3 that then can remove these serine ADP ribosylations again. But of course, what we also could see in our data sets, although we see a majority of serine ADP ribosylations, we also do see a little bit of other linkage types to other amino acids. And of course, it's not really known what exactly is catalyzing these types of linkage ADP ribosylation. But again, there's a plethora of enzymes out there that is not really well described yet and likely could be contributing to different types of linkage ADP ribosylation types. What is interesting is that when we look at the localization of these individual linkage types, ADP ribosylation, we actually do see kind of a localization effect. So you see serine ADP ribosylations is very strongly related to nuclear proteins, for example, whereas histidine ADP ribosylation very strongly relate to mitochondrial proteins. Arginine ADP ribosylations strongly relate to the plasma membrane, but also the endoplasmatic reticulum, while tyrosines very strongly relate to ribosomes, indicating that likely there could be kind of, very specific PARP enzymes that are present in these different types of organelles that is responsible for catalyzing these different types of linkage types. And of course, this is something that we hopefully can start exploring now that we have actually the methodology in hand to do these type of analysis in an unbiased manner. And finally, I would just also like to share some unpublished data with you. So one thing, of course, is the ability to study ADP ribosylation in cells. But of course, tissues is a completely different ballgame, and we have now started to venture into this as well. So we've done a small pilot study where we actually excised 11 different types of tissues from mice and analyzed the ADP ribosylation signature across those 11 types of tissues that were selected based upon at least literature reported aspects related to ADP ribosylation. And while I cannot share all the findings in detail, I can say that essentially what we did was we took 5 mice as 5 replicates, excised 11 tissues from each of them, and analyzed this using our ADP ribosylation enrichment strategy. We do see very good reproducibility across the different tissues and replicates, across the different replicates, whereas we do see tissue-specific differences across the different tissues, indicating likely that also different PARP enzymes might be active across the different tissues. Finally, just in summary, we have an enrichment strategy that is now can be widely used for studying ADP ribosylation. It can be used to provide, functional insights into PARP enzymes, as we have now demonstrated with new insights into the most widely enzyme, at least PARP1, but also likely applicable to many of the other PARPs. We can see HbF1, AR3 are inversely regulating the PARP1 catalyzed serine ADP ribosylation on a global scale. And also when we remove HbF1 or when we lose HbF1 in cells, we do not see a PARP1 specific switch to glutamic and aspartic acids. We just simply see that the enzyme primarily is catalytic dead. Also, for some of the other amino acids modifications or ADP ribosylation modification, we see strong spatial preferences that could indicate that it's individual enzymes in those organelles that might be responsible for catalyzing these types of linkage types. And finally, yes, now with the availability of doing these type of analysis in tissues, we can of course move into different types of mouse models and also disease models that hopefully also will provide further insights into the ADP ribosylation biology and ADP ribosylation dynamics. So with that, I would just like to acknowledge the people in my group who did actually the work presented here. So that was work preferentially by Sarah Larsen and also Ivo Hendricks, that are 2 postdocs in my lab, but also through our collaborators, the ARHEL lab, and also with the help from our local Lucas lab here at the CPR. And of course, also I would like to thank essentially the funding agency that has made all this work possible. So with that, I would just like to, thanks a lot for your attention.
Thanks a lot, Michael. Yes, clap. As I remind to all those who connected in the last minutes, is we keep the question and answer session later on. So we now, and please start write your question into the Q&A toolbar. In the meantime, we directly move to the second talk of this first section, which will be given by Irene Garcia from the University of Barcelona. She's going to...
No, sorry, I'm from Seville, from the CSIC.
Sorry, this was, okay, from Seville. You worked before in Barcelona? Or?
No, never.
Never, okay. From University
it's
of
a
Seville?
mistake that I don't know why.
It's
like
continuing.
but
I hope that the title is true. That is correct one. You
title
are
is
going
correct.
to talk about serine cyanidation in a different rain in plants. No.
No.
So then it's all a surprise.
Cyanide regulates cellular processes through a new post-translational modification of protein, S-cyanidation.
Ah, yes,
It's
which is what I said. Yeah, yeah.
Ah, I'm sorry, I didn't hear you.
So it's going to be cyanidation of serine in protein from plants. Here it's your the microphone is yours. Thanks a lot for being here.
Okay, everything is okay. Do you see my presentation?
It's not in presentation mode though. You should.
Strange. I don't know why. How is it? How do you see that? I'm sorry.
We see the slide. If you click the presentation mode
I already
down there.
did it, but I am, in this Macintosh or I don't know, something. Now I am in presentation in my computer. So it has to be something strange here. I don't know. I don't know how to. Maybe now. Is now correct?
Not yet.
Not yet.
But
well,
if you do not have animation, maybe we can go through the slide. This format.
Strange. Now? Is now correct? It's not in presentation.
No.
I can't do anything. I am sorry.
No, but we can see them clearly, so maybe.
Exactly. I think you can
so
use the slide in this format.
I just make this last attempt. Is it okay? No. It's not working. Okay. Well, so I just do it in that kind in not presentation mode. Okay, so I want to first of all I want to thank everyone, all the organizers here for inviting me to this meeting. I just want to start my presentation, but taking your attention the fact that world is warming up. Okay, so plant biologists mainly have an important challenge in this way. It's to obtain more food without using more land, without further damaging our planet and respecting biodiversity in a changing and warmer world. One of the strategies to fight against the climate change is to adapt to it. Plants are really the queens of adaptation, and that's well, plants have evolved very extremely efficient signaling processes or signaling processes for the integration and the detection of changes and to adapt to them. And that's why plants are sessile. They are not like animals. They cannot move from one place to another to escape from the changes or the stresses. The plant that we use for all of our studies is a model plant called Arabidopsis thaliana. It's like your mouse in animal systems, okay? And our question is how plant hydrogen cyanide is able to regulate processes. And our answer is through a new post-translational modification of proteins, which is cyanidation. This work has been done at the IBBF in Sevilla, Spain, not in Barcelona. All of you know cyanide is one of the most popular poisons in the world. Cyanide is toxic because it binds metalloproteins, as you can see here, in an irreversible way, including the cytochrome C oxidase of the mitochondrial electron transport chain. So cyanide, hydrogen cyanide, blocks the mitochondria function and leading to the death of the organism. Despite this, it is present in all the different, in all the kingdoms of life where function has been suggested or shown, okay? In plants, which are my organism, hydrogen cyanide is involved in biocontrol and in germination of seeds, but it has been normally considered mainly as a residue. Our question was, maybe hydrogen cyanide that shares a lot of properties with other signaling molecules can be also a signaling molecule. As you can see here, other small signaling molecules that are gasotransmitters, as NO, hydrogen peroxide, or hydrogen sulfide, have as cyanide as low molecular weight. They have very high chemical reactivity, and they are toxic at moderate concentrations, okay? But they have a very well-established signaling function at low concentration, and they all are gas or volatile, okay? So our question, as I told you before, is hydrogen cyanide as a signal molecule in plants. I'm going to convince you that maybe this is the question of the answer is yes. Cyanide is produced in plants during the biosynthesis of camalexin and also of the hormone ethylene. Ethylene is an important hormone in plants, and it is involved in the development and in the plant response to pathogens. Cyanide can be detoxified in plants mainly through the cyanoalanine synthase activity, the CAS. And in Arabidopsis, this is the major cyanide detoxification activity. Actually, in Arabidopsis, there exists only one CAS in mitochondria. There exist other activities outside of the mitochondria, but the mitochondria is the only one. And we have made use of a tool, important tool for us, which is the plant mutant in the CAS C1. Okay, these plants, the CAS mutants, show less CAS activity and more cyanide concentration in their tissues, going from 1.5 more than wild-type plants to 2 or 2.5, depending on the tissue and the condition of the plant, okay? Our work from the last, 10, 15 years have demonstrated that hydrogen cyanide is involved in root hair development and the immune response modulation in Arabidopsis, okay? Actually, the CAC1 mutant is more resistant to biotrophic pathogens such as bacteria and viruses. And this resistance is totally independent on a direct effect of hydrogen cyanide on bacteria, okay? This has been well established in our group, okay? Actually, one of the many things that we have shown is that very low levels of cyanides as 1 micromolar that are what we have more or less in our mutant plants are able to revert a mutation in the NADPH oxidase, which is one of the main proteins responsible of the pathway of response to pathogens that produce the resistance of plants to pathogen. When we mutate this NADPH oxidase, we find a very, very high sensitivity of the plant to the pathogen, as maybe you can see here, the infection of the leaf. When we treat the plants with 1 micromolar of cyanide, we see that this hypersensitivity goes down and the plant is more healthier than the wild type, okay? It is known that hydrogen cyanide prevents seed dormancy and stimulates germination through the reactive oxygen species production and protein post-translational modification as carbonylation, as you can see here. So we wanted to go further in this, and we wonder whether hydrogen cyanide was able or not to modify cysteine residues. I mean, the thiol group, which is a very reactive group in proteins, and it can be persulfitated, nitrosylated in reduced form, in oxidized form. And we like hypothesize that hydrogen cyanide could maybe modify the cysteine residue. It looked like a priori quite like crazy idea, but we adapt a method, an already existent method based in the fact that when you artificially cyanylate a cysteine residue, it makes the peptide bond close to that cysteine very sensitive to the cleavage with ammonium hydroxide, okay, and the protein is cut. So we ran several bidimensional gels, and we found a lot of differences between one and the other, the treated and the non-treated, and in that way we identified 93 possible cyanylated proteins. Then we moved to a more modern proteomics strategies by identification directly by LC-MS. Proteins that were cyanylated. We identified the cysteine residues that were that were more heavy, corresponding to the molecular weight of the cyanide residue. Okay, and in that way we identify 62 cyanylated proteins in plants, non-treated plants. Okay, and with all those proteins that were cyanylated, we did interactions network and groups. And interestingly, we found one of the groups that were more interesting for us was that one, the S-adenosylmethionine cycle proteins related to the DNA methylation, which are related to plant immunity because the methylation of DNA is related to the priming of plants to be like ready for stress. And this, I'm going to go back later to the priming state, okay? We have also done studies to look for cyanylation specific proteomes. Here I show you the mitochondria results. We identified 1,700 proteins. Among them, 11 were cyanylated via that modification. Several proteins of the immune response, oxidative stress. That is, was surprising because it was in root, but was like, was a little bit surprising, but normal. We also did proteomics in root hair. I remind you that plants, CIC1, are affected in the root hair development. So that's why we did this proteomic analysis of the root hair. Using the FACS strategy and proteomics, okay? And we identified in Phil Benfie's lab in the States 3,800 proteins. Among them, 49 were cyanylated, and among them, proteins involved in root hair development and the immune response, the oxidative stress, and so on. And what was very interesting for us is to find out, again, the proteins involved in the SAM cycle, in the S-adenosylmethionine and the DNA methylation process, okay? So now in this moment, we are interested in the proteomic analysis of the signaling mediated by cyanide in the plant microorganism interaction. And we want to, we are determining if there is a burst of hydrogen cyanide during the timing of the infection. I'll show you now. We are doing proteomics in the framework of the EPIC consortium at Petersman's lab. We want to identify signaling proteins in vitro expressed, the candidates, and study the activity, function, or localization of candidate proteins. So we are now centered in the proteomic analysis of the Pseudomonas aeruginosa infection, is a bacteria. And what we have shown time ago is that the accumulation of hydrogen cyanide increases quickly in the 3 first hours of the infection with the bacteria, and then it stays like that. And the CIC1 protein or gene is decreases, is inhibited, decreases the transcription at 3 hours after the infection. So we decided to do proteomics at that time. As I told you, at Peterman's lab, we identified 3,200 proteins, and when we look at the gene ontology of the groups of proteins, we can see a big difference between the wild-type behavior and the CIC1 behavior. CIC1 is the plant that have more hydrogen cyanide in normal conditions. Okay, so here in the wild-type we can see the several geogroups related to plant response to pathogens, okay, and the methylation, okay, but that we don't find in the CIC1 case, okay. So it looks like CIC1 does not feel very much like it is infected by a bacteria. So maybe it's CIC1 in a prime state, and here we go with the prime state. A prime state in our system is like a state in the plant where it is prepared for stress, okay? In that case,
sorry, you have 2 minutes.
Okay, I will finish now quickly. So the plants are like in a pre, they are ready for the stress, and it doesn't mean that that the plant is has less the fitness is slower. It's the same fitness, same productivity, everything. So that's why it's a very interesting state to study. Okay, and we are looking at several markers of the priming, scallop deposition, activation of malpinases, and DNA methylation. As I can show you here that DNA methylation is different in CSU1 mutant and in wild-type plants. This is something that we will never have seen, have studied without the proteomic analysis that we did. Okay. Going to the biological function of the cyanylation, we have already studied in vitro some the activity of the proteins subject to cyanide treatment, and we can see that in some cases the activity is lower in the presence of hydrogen cyanide, and sometimes the activity increases in the presence of hydrogen cyanide. So I think I can convince you that hydrogen cyanide can act as a signal molecule, even we have a physiological function. We have molecular targets and also a mechanism, which is the cyanylation. And this is not exclusive from plants. As other authors have seen that very small concentration of cyanide can stimulate mitochondrial function. And then we have started a collaboration with Sabasabo and Gary Boss to study more deeper the function regulation of hydrogen cyanide in living beings. So which is our contribution? Our contribution here is the implication of this cyanylation in the signaling ways to adapt for plants to adapt to environment, and in a long term maybe use it for improving the production. We have the tools and the knowledge, and they can be transferred to other sciences as biomedicine, biotechnology, microbiology, etc. I want to thank our research group and collaborators and funding. Thank you very much.
So thanks a lot, Irene. Due to time limits, we immediately move to the last talk of this session, which will be given by Albert Heck from the Utrecht University. I suppose this is correct. He's going to talk about protein histidine phosphorylation, to be or not to be, and maybe also a bit of the use of IMAC for XLMS. Let's see, the talk will tell us if we will hear about the two topics. So thanks a lot, Albert, and please, you can start. We can see your presentation in presentation mode. Thanks.
Perfect. And you can also hear me?
Yes, we can.
Thank you very much, Tiziana. Thanks everyone for attending. I'm glad to see that although we can physically meet again, that the interest into these webinars is still high. We are with more than 100 people, so that's a very good turn up. Thanks for also inviting me, Tiziana and Chris. I just will go quickly ahead and stop me when time is over. So I want to share some data that from our lab, where we work on protein phosphorylation, but also on how we enrich phosphopeptides. And you might say, okay, what's new about that? Maybe I first want to show that I go a little bit too fast. If we talk about protein phosphorylation, then in the years behind us, a lot of progress has been made. And now routinely, many labs can do phosphoproteomics. They use all kind of enrichment techniques for that. And the numbers of phosphosites that we can get from a cellular lysate, they are really astonishing. They are about more than 10,000 and sometimes even 40,000 or so. And this picture is a bit old, but it hasn't advanced much further than this. But you can say, okay, phosphoproteomics is now quite routine. And in many labs, and also many labs involved in EPIC-XS, it's now used in many studies. It's automated, and here you see our approach with a robot that we use to enrich for the phosphopeptides. And we can go to really low sample amounts that you see over here as well. You can go to 1 microgram or sometimes even less. So everything seems to be very well established, and people are using this sort of approaches now all the time. So today I want to talk about something else. If we talk about phosphorylation, we typically talk about serine, threonine, and to a lesser extent tyrosine phosphorylation. And a couple of years ago, we were intrigued, and you see by we, you mean I mean Simone Lemire, Clément Potel, and also Miao Xiaolin. We were intrigued by, is there other phosphorylation also playing a role in mammalian cells? And one of the suspects was histidine phosphorylation. So Clément set out to see if he could do this histidine phosphorylation. Now, we did it slightly differently than we did it before. We used a column that actually Simone developed, which is an IMAC column. It's very big column compared to what most people use with TIPS. And you can really load several milligrams of peptides on this column. And with this column, the input that you can give is very high. But then you can see that most of the peptides elute quite early, and you get a very sharp peak of your phosphopeptides. And this is really pure peak, and you can analyze this then by LCMS, and you really get a lot of phosphopeptides. Now, the nice thing about this approach is that you can do it under conditions that are not as harsh as the normal titanium dioxide tips. And for analyzing histidine phosphorylation, we thought we needed this because histidine phosphorylation, maybe a bit like the modification that Michael talks about, is even more labile than serine and threonine phosphorylation. So we thought we had to be very careful. Now, to make a very long story short, Clément and Miao used this approach, and they looked at phosphorylation in bacteria, in E. coli. And here you see the results of this approach. We were able to detect about 1,500 phosphosites in peptides. And to our surprise, and you see that here in the pie chart, to our surprise, we saw that most of them were serine, then also quite a few were threonine. But then of the histidine, we found about 250 of those, and even more than the tyrosine phosphorylation. So this was a good result, and we claimed that there was widespread bacterial protein histidine phosphorylation in bacteria. Now, I just want to say again, we could only do this by this specific approach, and we had very many controls in our method as well. And the nice controls that you can build in, and I just want to say that, you have of histidine, you have an ammonium ion, but of course also of phosphohistidine, you have an ammonium ion. So you can really look for this phosphohistidine ammonium ion, and that's a sort of verification that you really have a phosphorylated histidine peptide. Moreover, again, like Michael also said, because it's such a label modification, we had to use electron-based fragmentation techniques to do the sequencing of these phosphopeptides, these histidine phosphorylated peptides. But with that, we could really also get good site localization. So we had this tool in hand, and we published this a few years ago. And so we made the claim, and we were quite convincing, I think, that there is indeed histidine phosphorylation present in bacteria. But what now about histidine phosphorylation in mammalian cells? Now, here are some papers from over the last couple of years about histidine phosphorylation in mammalian cells. And it has been claimed that it's there, that it's actually quite ubiquitous. And even several groups have made now antibodies that are not targeting, let's say, phosphotyrosine peptides, but that can really target phosphohistidine proteins. You can use these antibodies to pull down these proteins, and then you can analyze by proteomics these proteins, and you get a view of the phosphohistidine phosphorylated proteins. Moreover, there's also a paper where they really did a large-scale phosphoproteomics, used many enzymes, many proteases, and also there it was reported that there were quite a few histidine phosphorylations in human cells. So in our lab, we had a PhD student, Niels, and we asked him the question, you see the difference in the slide, are there really histidine phosphorylations in mammalian cells? So here's Niels. He started about 4 or 5 years ago, and he worked on this throughout his PhD. Now, the first results that he obtained was this normal workflow that I showed you before. And he analyzed here 4 different mammalian cells to quite some replicates. And for all these cell lines, he found, you see the numbers, about 25,000 up to 30,000 phosphorylation sites. Of those, most of them were again serine, as you expect in a mammalian cell line. Also quite a few threonine, but again, the histidine looked quite high, up to about 1,000 histidine phosphorylated peptides, quite higher than phosphotyrosine. So this was his initial result. As for many PhD students, you get excited, you think it's all brilliant, but luckily Niels and Simone started to look more critical at that data. And one of the things that they noticed, that if they looked at site localization and used the scoring in MaxQuant, for instance, and looked then at the sites that were defined as class 1 phosphosites, so the ones that are better localization scores, they actually saw that the number of serine sites dropped by about 20 to 30%. The number of threonine and tyrosine also dropped by 30 or 40%, but the number of histidine, which you see here in red, really went down by 80% or so. So it turned out that site localization, if you made that more stringent, you lost quite a few of your histidine phosphorylated peptides. So this was already one thing that we thought, okay, what's going on here? Is it maybe something bizarre? Now, the other thing we did, and that's shown over here, we looked how many of these sites are actually observed in all the 4 different cell lines. And of course, this doesn't need to be high. They are different cell lines with different proteins. But typically we find that about 30 to 40% or 30 to 20% of the sites are retained from one cell line to the other cell line. But with histidine, this number also dropped to very low numbers. So this made us think that this histidine was a bit weird. Now, then we did several experiments, and this took them really several years. We also thought, okay, it's known that histidine phosphorylation is actually acid labile. So what he did is he took his whole pool of phosphopeptides and acidified it for a while. And then indeed in E. coli, we saw a dramatic decrease in the amount of histidine phosphorylated peptides. You see that over here. But in the mammalian cells, if we acidified them, it almost had no effect on the number of phosphopeptides that we detected. So this sort of shows, and it's particular that the histidine sites are more labile than the other sites, and that's why we plotted this ratio. It again made it suspective if these histidine phosphorylated peptides were indeed histidine phosphorylated peptides. Now, next thing that we did, and also Michael showed plots like this, we looked at the localization probability of all the peptides. Now, the localization probability is a score on how well you know that the site is really phosphorylated on that particular amino acid. Now, you see that for serine, you get quite a nice localization probability profile. For threonine and tyrosine, this is already a little bit less, but it's still okay. And then for histidine, we really see that this localization score is as bad as I think Mikkel showed before for the DNE amino acids. So it really shows that the localization score for histidine phosphorylate peptides was pretty bad. Now then we did a bioinformatics experiment where we said, okay, let's now also think that maybe we look for proline phosphorylation. And of course, proline phosphorylation is not possible. But if you allow it in your searches, you actually find quite a few proline phosphorylated peptides. And of course, this is because then maybe the proline is not phosphorylated, but the amino acid adjacent to the proline is phosphorylated. And if you don't have fragment ions that can distinguish those, actually your software cannot distinguish whether it's phosphorylated on the proline or the serine. So if we did this, we found a proline localization score, which was as good as the histidine phosphorylation score. And then, of course, if we looked at the proline phosphorylations, then almost always the adjacent amino acid was a serine. And that serine was then phosphorylated instead of the proline. If we did the same with the histidines, we actually also observed that most of our histidine phosphorylated peptides that we had assigned had neighboring serines or sometimes a threonine. So we actually think that all the sites that we initially thought to be histidine phosphorylated turned out to be not histidine phosphorylated, but turned out to be adjacent phosphorylated and then most likely on the serine. Now we did many more tests. We also looked for the ammonium ion. We looked for other reporter ions. But to summarize this story, we started in E. coli with about 150 histidine phosphorylated sites. If we looked then at class 1, if we looked at the acidification, if we looked at the ammonium, we still could retrieve by this very stringent filtering about a fifth or a sixth of all the phosphosites. If we did the same with the human cells and then do the class 1 filtering, the acidification and the ammonium ions, our numbers from 1050 dropped to 2. So this made us think that there is actually no real human histidine phosphorylation, but there are exceptions, and the exceptions are here at the bottom. These are the ones we find, and it's quite interesting because the only ones that we find are not histidine phosphorylations due to a kinase or a phosphatase action, but they are known intermediates in metabolic reactions. So that we detect them means that we can detect histidine phosphorylation, but that we don't detect any other ones made us now really think that there is no histidine phosphorylation in mammalian cells. So that's summarized over here. Out of the 1200 histidine peptides that we initially saw, by using already quite stringent filtering, only 4 of them were left over when we did all the other things that I mentioned. So Tiziana, I look at you now, and how many more minutes or seconds do I have?
I think you have 2 minutes.
I will see what I can do. So then I will stop in 2 minutes at least. So this is the first part. The second part is, that this phospho enrichment works so well. In our lab, we have started to think about how can we explore this now in many more ways. And the first one I want to show, I think you might know already. This is where we actually used a phosphonate tag on cross-linked peptides. And just to tell you the story, if you do normally analysis of cross-linked peptides, it's extremely difficult because they are in the background of maybe hundreds more or thousands more or millions more of normal peptides. So typically when we did cross-linking mass spectrometry, we did very deep fractionation in the hope to find also some cross-linked peptides. And here in this diagram, you see then of all the peptides that we detect in our analysis, about 99.9% were normal peptides. And if we are lucky, then maybe 1% or maybe even less comes from the cross-linked peptides. So cross-linked peptides were normally really a needle in the haystack. And then Barbara and Richard in the group, they developed this FOX cross-linker. And here you see what we are using. So this is not a phosphate, it's a phosphonate. And we have here the warheads that can couple to the lysines. Here we have the phosphonate that can bind to the titanium dioxide or the iron IMAC columns. And we use that now to enrich the phosphonate cross-linked peptides from a whole cell lysate. Now, and I tell this very quickly. So here you see the initial Venn diagram that I showed you before. And here you have all the normal peptides and just very few of the cross-linked peptides. If we do this now and we do again use the same phospho setup as we use for phosphopeptides, we can actually come to an analysis where the normal peptides are now 1E8, the phospho cross-linked peptides are 2E9. We also still have some monolink peptides. So it again shows that this enrichment that we use for phosphopeptides can also be used for phosphonate linked peptides. And we did this for cross-linking. You get very nice spectra with specific precursor ions, but I won't go into much detail because this has all been published. But I want to acknowledge the people that did all this work. And very quickly, and really as a last thing, nowadays in our lab, several people are working, can we maybe replace the biotin that is so popular in many applications, can we maybe also replace that by a phosphonate? And so here you see a method that's nowadays quite popular, BONCAT, with all kind of variations, where you actually use this AHA labeling to maybe measure secreted protein or measure newly synthesized proteins. And then you can click your biotin to this alanine replacement amino acid, and you can fish out for them. Now in our lab, we developed, and Fleur did this with Barbara, we developed now an a variant that then introduces the phosphonate to that, and then we can use again the same platform. And now we can enrich for all the ones that contain the AHA label with the phosphonate handle. And we can also start to analyze these quite easily. And it really works very well to look for newly synthesized proteins that you pulse in the AHA compound at the same time that you want to study this. And you get really very clean spectra for doing this as well. So I think I want to end there, just say that enrichment of phosphopeptides is maybe now routine and quite easy, but that doesn't mean that we know everything about histidine, about phosphorylation, for instance, histidine phosphorylation. But I hope I can also show you that you can use these IMAC columns also for other applications, and maybe also for chemical proteomics, as I showed here in the slides as well. So with that, this was published, and here's the publication. We benchmarked it against biotin for PhosID versus BONCAT. This was really driven by Fleur, Barbara, and Wei Wu in the lab. And if you're interested in that, you can have a look at that presentation. So with that, I'd like to end and thank everyone for your attention and thank the group for their support. And with that, I give the word back to you, Tiziana. Thank you very much.
Okay. Thanks a lot, Albert. Thanks to all the speakers. I think we can start with the Q&A session again, because there were many people just logging in during this first session. I remind you, you can write your question in the Q&A toolbar in Teams, also in Anonymous, or you can raise your hand. Here, Chris, I ask your also help. We can read one question after the other, and maybe we can go for one question, the first round of questions to all the speakers. So I can start with the first one for Michael. Is a question from Chris, indeed. Have you already tested the EAD Cyax-Xeno-TOF on ADP ribosylated peptide?
Yes, thanks, Chris. That's a phenomenal question. No, unfortunately, we have not had the possibility of doing that. I've been in contact with Cyax, but it would be very exciting to test the new EAD capabilities of that instrument, definitely. But so far, unfortunately, no luck.
Chris, you want to ask a question?
Yeah, I'll ask August's question then to Michael. How much is actually known, or how much is there mono and poly ADP ribosylation? Can this be quantified? Is it more difficult to quantify poly ribosylation?
From a mass spec point of view, definitely it's more challenging. So the theory goes that the PARP1 can catalyze this poly ADP ribosylation in up to hundreds of chain length, but people are starting to realize that maybe this is actually not what is happening after all. So several publications have been out recently, and we have also tried to explore this ourselves. And our data indicates that at least at the time where we are sampling the cells, we see primarily only mono ADP ribosylation. But again, the modification is so dynamic and usually activated within seconds, so it can easily be that the poly ADP ribosylation is generated very rapidly and then very rapidly also turned into mono ADP ribosylation where we are sampling it. But they are kind of becoming a more general consensus that maybe it is more like mono versus oligo ADP ribosylation than necessarily these long hundreds moieties of ADP ribosylation that is part of this modification. But there's still many open questions, and yes, quantifying poly ADP ribosylation by mass spec is of course challenging.
Okay, so we have a question by me to Irene. So Irene, you show different levels of cyanylation in different functional state. So you show this as a signaling molecule. So the question is, do you have evidence that there is an enzymatic regulation of this, or it's only a chemical? And if so, how do you see this PTM to be regulated or modulated? You are muted.
It's true, sorry. We don't have any evidence of an enzymatic modulation of the modification. We don't have any data. We think that it is like the nitrogen oxide or hydrogen sulfide case. It's a chemical thing. But we don't have any. This is a very recent modification. We have very few data. And the other question was?
No, if you have an idea how it could be modulated, if it's not like pushing degradation of the protein or destabilizing
we don't have any evidence, but we think that it depends on the amount of hydrogen cyanide that is in the cell in the moment in the area where the protein are. But we don't have any data yet.
Okay.
It's difficult to,
Thanks.
we're working on that.
So Chris, you have a question?
Michael, there's another question for you from Tiziana. So I'm kind of like the postman here. And she wants to know the use of the macrodomains. How does that compare to, I guess, other affinity approaches, such as tagging or antibodies?
Yeah, so we have actually tested that quite extensively in the beginning. So there are different types of ADP ribosylation binding domains available, but when we compared those against each other, we found the AF1521 to be far more sensitive and specific. And also we've recently tested actually against commercially available antibodies from cell signaling towards ADP ribosylation, and also in our hands, the AF1521, the natural ADP ribose binding domain is far more sensitive and specific than the actual antibodies.
Thanks. So I see that there is one raised hand. So it's from Juan C. Royas. So Martina, if you manage to switch on the microphone. In the meantime, I can ask a quick question to Albert. So what about the histidine phosphorylation that have been annotated on histones? Because there was quite a bit of literature about them.
Yeah, so I think we have some doubts about it. We don't see it back, but of course, you never know for sure. And the problem that we have seen is that all the histidine phosphorylated sites that have been reported, they always have serines adjacent to them. So to give really an answer, I have to say, show me the spectrum and let's manually interpret if it's really on the histidine or if it's on the adjacent site. And if you can really convince us that it's on the histidine, yeah, then it might still be there. But the trouble is also there's no kinase, there's no phosphatase, so then you have to think about how does this happen. So in the paper we also don't say it's never there, but it's certainly not playing a big role in mammalian signaling.
Hi, can you hear me?
Yes.
Okay, so again, my question is to Albert because it's kind of related on this topic. I am amazed by how many possible hits were proposed for a histidine phosphorylation that after you have a look at it, it was all discriminated. And as someone that has worked at PTM for quite a few years, it's kind of alarming. I always like looking at my spectra before saying, one, this is the PTM I have, and two, this is where I think it is, because I'm always very skeptical of the site localization algorithms. And I think this was a clear example of we should maybe pay more attention to this. So it's a kind of a general question, but where do you stand in this? Like, how do you think we should move forward? Yeah. This is phosphorylation. It's one of the most studied PTMs, not even to mention how good the algorithms will be for localizing for other type of post-translational modifications.
No, Juan, I think that's a very relevant question, and I'm actually glad you asked this, and maybe also later on Leonard wants to comment on this, or even with serine phosphorylation, you should be very skeptical or very critical if you have phosphopeptides where you also see that there are 4 or 5 serines in the peptide, and this is not unusual. And I think also phosphorylation happens a lot in serine-rich stretches on proteins. So you're pretty sure that there is a phosphorylation there then, but if you can say if it is on serine 166 or 167 or 168, yeah, you really need to have the right fragment ions to make that discrimination call. And also in that sense, I think a lot of the high throughput data sets, yeah, if you really critically evaluate them, then I'm pretty sure that we are now pretty good in saying this is a phosphopeptide. But for saying this is the site, yeah, that really depends on the context of that site that you want to report on, and that is something where I think we as a community can still improve a lot on.
Thanks.
Thanks a lot. So Martina, what do you think about timing? Because there were a few questions still posted, but maybe we are short in time.
Yeah, I think we're short in time, and we had promised already a coffee break.
Okay. So then we can close this first session. I would like to thank very much the speaker once again. We have a 5 minutes break just to for a quick refresh. So we meet at 4:20 to start the second one. So thanks a lot. See you.
Thank to you.
Hi, Neil. I see you're already here. I've just sent you an email. I don't know if you can hear me. Hi, Tiziana. I see Neil is here, but I don't know if he can hear me. Or maybe he's gone for a break.
Maybe, yeah. He's in the list.
He is here, yeah. I've just sent him an email and I just
Okay.
see him now. Well, I'd gone through his slides yesterday anyway, so I'm sure everything will work out fine.
Mhm. I think maybe in the interest of time, we better start again.
I think
Yeah,
so.
I think so.
Okay. So welcome back, everybody. I hope you all got a espresso rather than probably a large coffee. So I think we're ready for the second session of today. I'm going to give the word to Mariana Maniaci from the IEO. I hope I pronounce your name correctly. I will stop sharing my screen now and give the floor to you, Mariana.
Thank you so much. I hope that you see my screen now.
Yes, not in presentation mode though.
Perfect.
Yeah, okay.
Okay, thank you so much. And first of all, I would like to thank all the organizers to give me the opportunity to present my work today. And today I would like to talk to you about the role of the arginine methylation in the adaptive response of cancer cells to cisplatin treatment. Protein arginine methyltransferases, or PMTs, are the enzymes that catalyze these PTMs. And in mammals, have been identifying 9 different types of PMTs. PMTs can be divided into 3 different types, depending on their capability to put just one monomethyl group on the guanidinous group of the arginine, or to put one monomethyl group and then a second one in asymmetric manner, and in this case are named type 1 PMTs, or in symmetric manner are the type 2 PMTs. Among them, PMT1 and PMT5 are the most active of PMT type 1 and type 2, respectively. Arginine methylation is an important post-translational modification because it's involved in a lot of biological processes, such as the regulation of the transcription, both in terms of activation or repression, depending on the site in which it's added, or in the regulation of the splicing process, and in particular in the assembly of the splicing machinery, and also in the regulation of the NIRNA biogenesis. Another important feature is that the modulation of the arginine methylation and the expression of PMTs in cancer is frequently overexpressed. And for this reason, a lot of inhibitors have been designed to use as a strategy to selectively reduce the tumor growth. Moreover, it has been shown that in some type of cancer, the combination of different type of PMTs inhibitor could also synergize the effect of the anti-tumor effect. For this reason, we start to study the modulation of the arginine methylation in the context of ovarian cancer. And from literature, we know that platinum-based treatment is the major type of treatment for this type of cancer, and that commonly the resistance to cisplatin treatment can occur. And so we design a proteomic approach that includes SILAC metabolic labeling of the cells to compare two different conditions, the untreated condition versus the cisplatin one. Then a common workflow from the proteomic expert. So the in solution by trypsin of the protein, then the HPH reverse phase fractionation of the peptide, and in parallel directly mass spectrometry analysis for the identification of a total proteome, and the immune affinity enrichment of R-methyl peptides for specific identify the so-called R-methyl proteome. Of course, the experiment was performed in 2 replicates, forward and reverse. And when we plot the different SILAC ratio to compare the cisplatin treatment versus the untreated condition, we identify a group of R-methyl peptide that seems to be strongly downregulated after the treatment with cisplatin. And we notice that most of them, around 70%, belong to protein named RNA binding protein, and that most of them, more than 50%, are already validated to be component of the so-called membraneless organelles that are granules that usually forms dynamically in the cells as a consequence of a stimulus, and that are composed by RNA binding proteins and RNA. Starting from this observation, we noticed that PRMT1 seems to accumulate into the nucleus after cisplatin treatment, and this relocalization of PRMT1 could lead of a sort of hypomethylation of some targets in the cytosol, and that most of them indeed seems to be RNA binding protein. So we was wondering if this modulation of the arginine methylation on RNA binding protein could in some way impact on their RNA binding capability. To evaluate this possibility, we design a specific proteomic approach, again with the use of Scilab metabolic labeling, and in this case, the pharmacological inhibition of the two major PRNTs. So the use of MS023 as inhibitor of PRNT1 and GSK 591 as inhibitor of PRNT5, and conjugate this with a particular strategy named OOPS, or orthogonal organic phase separation, that thanks to the physical chemical properties permit to identify specifically the complex formed by RNA binding protein, or RBP, and RNA. In particular, this protocol required the UV cross-linking to fix the binding between the protein and the RNA, and then thanks to a triazole chloroform mixture and a serial centrifugation, we can distinguish three different phases: an upper part in which there are the free RNA, a lower part, the organic phase, in which there are only the free protein, and a small interface that are enriched for these MBP's RNA complexes. Then, for our purposes, we digest this interface with RNA and analyze in parallel with a small fraction of whole cell extract the differential intensity of the protein, like a normal workflow. So in solution digestion by trypsin, and again HPH space fractionation prior to LC-MS analysis. From this type of approach, we can see two things. First of all, that the PRMT5 inhibitor have not significantly changed in terms of RNA binding capability, so seems to not really impact on this biological process. While for what concerns the inhibitor of PRMT1, we can identify in the interface a group of proteins that seems to be strongly upregulated. So this means that should increase their RNA binding capability, or at least enriched in terms of NBP's RNA complexes. When we compare with the total extracts of whole cell extracts, we don't see the concomitant type of enrichment. So this means that the effect in the interface are really due to a change to RNA binding capability, and is not just the mere reflection of change at the level of protein expression. To better highlight this difference, we decide to normalize directly the ratio Silac into the interface over the ratio Silac in the whole cell extract, both for the forward and reverse experiment. And as you can see here, again, the group of proteins strongly upregulated emerged. Now we know from literature that for single RNA binding protein, the regulation of the arginine methylation in some way change also their subcellular localization and possible formation of this type of granules. For this reason, we decide to set up immunofluorescence screening, in which we compare the protein of interest, in this case LDHP, and possible co-localization with the G3BP1 as marker for the stress granules, and obviously with RNA. And as you can see, after the treatment with PRMT1 inhibitor, we can observe the formation of these granules in the cytosol that perfectly co-localize both with the G3BP1 and with RNA. And then as control, we decide to use the U1.6 hexanediol, that is an alcohol commonly used to dissolve this type of granules. And indeed, there are no more formation of granule, even if we treat before the cells with PRMT1 inhibitor. As expected from mass spectrometry analysis, no particular change can be observed after the inhibition of PRNT5, and as future control, we decided to use the sodium arsenide, that is a oxidative type of stress commonly known to induce the formation of these granules. And again, you can see they are quite similar to that one formed after the inhibition of PRNT1. Now that we have demonstrated that the modulation of the arginine methylation, that in particular the modulation of the balance between the asymmetric demethylation and the monomethylation have an effect on the RNA binding capability, we come back with our workflow and in this case compare the untreated condition versus the cisplatin treated condition. You can see for different replicates because in this case are in label-free condition. Again, the hoop strategy, the protein digestion and peptide fractionation, and the obtained results have been normalized as before over the full cell extract in order to, again, highlight better the difference due to RNA binding capability changes and not to a mere reflection of the change at the level of protein expression. And as you can see, when we compare cisplatin versus untreated condition, there are still a lot of protein that seems dynamically regulated by cisplatin. For this reason, we start to focus on that protein that seems to be enriched in the interface, so to bind more the RNA. And we notice from gene ontologies analysis that most of them are indeed ribosomal protein, and that seems to target the endoplasmatic reticulum. Moreover, since our initial hypothesis was to link the effect of cisplatin directly on methylation, we try to intersect the list of this protein that seems to be dynamically regulated after cisplatin treatment with the list of protein annotated as arginine methylated in both our in-house high confidence R-methyl database named Prometheus DB or in the online available Phosphosite Plus. And you can see that most of them indeed are at least annotated to be arginine methylated. Now, the final goal will be to demonstrate that this R-methyl site could be in some way modulated after cisplatin treatment, and if so, if they are really crucial to their RNA binding capability. And to do this, we would like to map in depth the possible dynamic changing R-methyl sites and then validate by amino acid site mutation the importance of the arginine methylation. So our initial model is a little bit more complex. So we know that PRNT1 seems to accumulate on chromatin and activate transcriptional pathway, but in parallel we have not forgot the cytosolic compartment, which seems that specific type of RNA binding protein, in most of the case ribosomal protein, seems to be seems to result arginine hypomethylated. And as I said before, our initial goal is to identify the R-methyl sites that dynamically change after the cisplatin treatment. And since we saw before by immunofluorescence that the modulation of the arginine methylation induced the formation of the stress granules, we would like to see if after cisplatin treatment we can observe similar type of granules. And moreover, since we saw from gene ontologies analysis that this ribosomal protein seems to in some way target the reticulum endoplasmatic, we would like to perform co-localization analysis with some marker for the endoplasmatic reticulum. And just really as a preliminary results, we start with the first part. So to identify potential interesting and dynamically modulated R-methyl site after cisplatin, I performed the immunoprecipitation following mass spectrometry analysis on one of that ribosomal protein. And you can see, of course, just one more just the Western blot control to see the enrichment of the protein, and in parallel the in-gel digestion with trypsin prior to high resolution mass spectrometry, and this leads to the identification of a new methyl site only in the untreated condition that had not been previously annotated on FONSP site or in other type of publication. So actually we would like to validate also other possible target in the same manner. And with this, I would like just to thank all the group, the component of the Tiziana Bonaldi's group, of the component of the mass spectrometry unit, and of course all of you for the attention.
Okay, thank you very much, Mariana. I just admit somebody else here. Just as a reminder, please do use the Q&A button at the right, in my case at least, the right top of the screen here, to ask or to write your questions, and we will compile them afterwards, so after the talk of Leonard and Neil. So I kind of like already introduced Leonard. So Leonard, you can take control of the screen, and that will be more like a bioinformatics talk, I assume. And I think the word IonBot is somewhere missing in your title. Leonard, the stage is yours.
Yep, the word is missing from the title, but it will not be missing from the presentation, don't worry. Okay, I'm just getting my presentation here, and you should be seeing my slide.
Yes, we do.
We do.
Okay, super. So I'm going to talk a little bit about an unbiased exploration of the proteome-wide modification landscape. So we've talked in this nice seminar a lot about specific modifications, but here we're going to try and do an unbiased exploration, because one thing is very clear, is that there are many modifications out there and that we don't know them all yet. Now, the thing that I take home from all of this exploration that we have been doing in my group is that it's very much like dinosaurs, in the sense that when I was a kid, this is how people thought about dinosaurs, including me. So they were very lizard-like, horrible creatures that were fantastically entertaining because they were frightening and strong and big and inspiring. But you also notice that they are very scaly, and usually they were green or gray. Now, something happened in the interim, and people found new fossils, and it turns out that this is not really what many dinosaurs looked like. In fact, this is what dinosaurs look like today. So they have feathers, and they look like strange chickens. They still have teeth, though. That's the one thing, and claws. But for me, this was a very big shock, because you have to completely alter the way you've thought about these animals all your life, and it actually comes as a literal shock, because you have a hard time accepting this change in a way. Why am I telling you this? Because I think proteins are going to go the same way. I think we underestimate the complexity of the proteome. We underestimate the impact of PTMs, just how many there are, just how many there could be on proteins, and just how much information is lying there that we haven't been actively using yet. And we've had that kind of as a message so far already quite extensively. I think I'm just going to try and hammer that point home even further. The good news is that whenever you see a transition like this, you are actually in a very interesting time in the field, and there's lots and lots of opportunity for young people who are nimble and who are fast and who can get on this train of new discovery. So I will talk very briefly about how we built the tools that allow us to do proteome-wide modification analysis, and we do that by using machine learning tools that learn how our analytes behave in the instruments. And then, as Chris mentioned, we built a new search engine called IonBot, and I'll use a slide to introduce that thing, and then I'll tell you what happens when you apply IonBot for proteome-wide PTM discovery on a very large amount of data. So very briefly, how do we got started in all of this? We started building a very serious spectrum predictor, MS2PIP, which came out in 2013. So it's been around for a while. It was one of these early efforts in this field, and we've kept evolving it ever since. And so it's a pretty good model. It allows us to predict spectra quite accurately. The things at the bottom show you that here it depends on which instrument you use, how good you can make this. And it also, of course, means that if you have a model, say, for HCD, and you apply the HCD model here in orange, if you apply that to, say, TMT data, it doesn't perform so well. But if you build a specific TMT model, it performs much better. And you can see here why. The HCD model prediction is on top. The real TMT labeled peptide spectrum is at the bottom. You can see that actually there's a lot of peaks missing and the intensities are very different. But if you have a TMT model, the TMT model actually predicts the spectrum really well. And this is something that we already published in 2019, and the model has been available since then. So this is one aspect of the behavior of our analytes in the mass spec that we can predict. We can predict what the MS2 spectrum will look like. The other thing that we can predict is a retention time. So retention time predictors obviously have been around for a very, very long time. But we built one called DeepLC that is very different in the sense that it can take into account modifications and the effect of that modification on the peptide retention time, even though you have never trained it for that specific modification. So it can actually learn how to predict the effect of a modification on the retention time, even though it has never seen that modification. And you can see that when you predict the retention times of a bunch of peptides that have been acetylated, but with a classical retention time predictor, what you will see is that the observed retention time is always larger than the predicted retention time because the acetylation makes the peptide more hydrophobic and shifts it back on the gradient. But as the algorithm doesn't know about acetylation, it will predict an earlier retention time. So there is a shift. DeepLC, however, despite the fact that in this particular iteration of training it, we never showed it an acetylation, so it has never seen one, it can predict very accurate retention times. That's the blue dots, which move away from this shift and go straight back onto the diagonal. The other thing that you should notice is that the width of the distribution changes. So the error on the predictions becomes smaller in DeepLC. And that goes for all these oscillations here, actually. And here you can see an overview of the effect of DeepLC across a whole bunch of modifications. Usually DeepLC does better, but not always. So there's a nice example here with phosphorylation, where if you never show DeepLC any phosphorylation at all, it has trouble figuring out what phosphorylation will do to the peptide. So it gets a little bit worse than when you don't know what phosphorylation is at all and you don't care. The reason for this is that phosphorylation, of course, is a bit of a special modification. It contains a phosphorus and 4 oxygens or 3 oxygens extra. And as a result, it's very difficult to predict a phosphol when you have never seen anything looking like a phosphol. And so that difference, that thing shows you that A, the algorithm is not magic, which is what you would expect, and it cannot do things that are completely crazy. But also it helps if you have more input, because the more variety you give the learning algorithm, so i.e. including phosphorylated peptides in the training set, the more likely it will be that it can predict an even broader range of modifications. So fundamentally, we try to feed DeepLC as much modified data as possible to get optimal predictions. And then that actually works out well because, of course, as soon as you introduce phosphorylation in the training set, this blue box looks a lot more like that one. And then things will be much better with DeepLC. So this is quite new and quite interesting because, of course, now we can use this to identify modified peptides as well, because we can check whether the modified peptide has the correct retention time. Now, how do we use that? Well, we built a search engine called IronBot. So what IronBot does is it takes this fragmentation prediction from the MS2PIP algorithm that we built and the DeepLC retention time predictions, and it's taking all of these predictions for all of the candidate peptides and then builds a fully machine learning-based scoring system on the fly. So there is no more any predefined scoring system here. It really learns from your data. And one of the benefits of having all of these predictions is that IronBot allows you to identify modified peptides. So why is that important? Well, if you look at IronBot competing with itself, you can see for 5 different public domain datasets that we just picked at random from the database, you can see that IronBot without open modification search does a good job, but IronBot with modifications will actually give you many more results, typically. So here the difference is not so big on this breast cancer data set, but on all other data sets, there is a huge impact when you allow open modifications. You can also see here that there's a TMT data set included. That's obvious because we've had the TMT model for so long. Now, if you zoom in, let's take this HEC239 cell because it's the biggest data set. What you can see is that the intersection between the blue and the orange is quite large. That's good news. So they overlap, they agree on a lot of the identifications. Then obviously the open modification search has many new hits in yellow and green here. That's all this orange bit on this chart. But there's also some modifications that are changed. So from the original blue identifications, several of them, so the majority in fact, are now removed by IronBot, and it says these are no longer correct. Some of them are just gone, but many of them have been changed to a new identification. And then the question is this new identification actually better than the old one? And the way you validate that is by looking, for instance, at the retention time, which again, DeepLC can predict relatively accurately. And here you see the retention times of those that are only found in the closed search or those that are overruled in the open modification search. And you can see that the retention times are really bad. So the predictions don't look like the real retention times. However, if you look at the ones that are overruled, the new identifications that carry modifications, they actually align beautifully where you expect them. So there is in fact something going on that these modifications, by looking for them, we can actually correct, let's say, good enough identifications that are actually wrong because we never considered the real identification, which is a modified peptide. So that was already a very interesting thing to find. And so modified peptides are important to consider because some of them are yielding very nice spectra that we are unfortunately matching to the wrong things if we don't consider the actual modified peptide. Now, this is all well and good, but what does it actually turn up? What can you find if you search proteomes? And this is the proteome-wide thing. So if you search in an unbiased way for modifications, that's what IronBot delivers. You can do that on a proteome-wide scale by taking a sufficiently large amount of data. So here I was talking about 5 data sets that we took from the public domain, but why stop at 5? So we can take many more data sets. And the first thing that we actually did was we focused on phosphoproteomics data, which is about 2,000 raw files, 60 million spectra, so a very small amount of data. 1,500 unimod modifications are considered by IronBot, as well as all possible amino acid modifications. And the results of that are given here, where we have about 19 million PSMs. We have about 140,000 P sites, which is reduced to 94,000 if we take a localization score with phospho-RS of more than 0.5. And there's about 14,000 phosphoproteins involved. And we named that resource Scope 3P, and you can surf to that resource and look at it today. And what it will show you for your protein is it will show you the protein sequence with all known phosphorylation sites. And then it will map these onto the 3D structure, which is interactively viewable on the website. Whenever a phosphosite is blue, it means it has a location on the 3D structure. When it's red, it means we don't have structure for that bit of the sequence, which is typically the termini here. Very recent innovation, by the way, is that we have all the AlphaFold structures included in Scope 3P, which means that now essentially every protein has a structure, an AlphaFold structure, but it can also have one or more experimental structures if these are available, of course. And then the other thing is that we have full provenance of the data. You can find out where all the data comes from. And we annotate all the biophysical properties. So the phosphosites are in the inner circle, and then you have lots of different biophysical properties that are annotated for each of these sites, which is really great if you want to do some machine learning on why these phosphorylation sites occur on these spaces. So this is very nice, and it worked very well. And we thought, okay, we can do this. So now why stop at phosphorylation? So the idea is let's take a lot more data, 25,000 raw files, about 1 billion spectra, all taken from the Pride database, run them through IronBot, and what do we get? We get about 215 million PSMs, about 740,000 peptides, mapping against 99% of the proteome, 20,000 proteins. 444 out of these 1,500 modifications are actually found, and they affect about 4.9 million modified residues. So you find a lot, which is essentially the summary. And then the idea was simple. You know what? We will do the same thing we have done for Scope3P, this phosphorylation database. We put the sequence, and then we put the modifications on the sequence, and then we put the modifications on the 3D structure and in the biophysical properties. And Bob's your uncle. We have a beautiful web system, and everybody can look at this data. Now, what you're looking at here is not real data. This is a mock-up, which you can also tell from the typo that is obviously left in the caption here. But when we looked at the real data, we were in for a little bit of a shock, because the real data looks like this. This is a random protein. We did not pick a particularly nasty looking protein. This is really just a random protein. And what you can see is for every residue, you can see all the modifications as colored circles. And what you can hopefully realize at this point is that proteins are a lot more complex than we thought. And now I come back to my dinosaurs that I bored you with at the beginning, because you can see that instead of scaly, almost quote unquote naked lizards, what we see now is something that is very much covered in feathers, covered in modifications. Keep in mind, however, that we're not looking at a single molecule. We're looking at all of the versions of that protein across all of the data that we accumulated in the PRIDE repository. So what we're looking at is modification potential rather than actual modification state. In fact, it's obvious because many residues have multiple modifications highlighted, and they cannot all be there at once, of course. Now, if you zoom in, it becomes a little bit more easy to deal with, and we're looking into doing better visualizations. We actually have a few suggestions now, so hopefully the system will be online soon for you to browse. But it's very interesting to see that certain residues carry modifications, but depending on where they are, the modifications that they can carry differ quite dramatically. So it's not that you're an arginine, you will always carry the same kind of modifications. It depends on which arginine you are, and where you are in the sequence, which kind of modifications you will carry. If you look at these modifications in more detail, and I don't really have the time to go into any of that detail today, but we can see that they are related to metabolism and chemistry of metabolism. They're related to the environment, so we can see environmental exposure on proteins, and obviously we can see regulatory processes in action. Think phosphorylation as the prime example. Now, this is a very interesting and very nice resource, and it comes with certain caveats, of course, because we've heard from several presenters already that we have to take care of the localization. We have to take care of verifying that all of these things are correct, and we are very heavily working at localization of PTMs in even more interesting ways than are currently being used. So better than, say, A-score or PhosphoRS. We have some nice candidates there, also based on machine learning. We just have to finish up implementing these things, writing them up nicely, and then chucking them into the Scope3P system. But Scope3P already has this PhosphoRS score that is then generalized to the PhosphoPTM score. So it's actually quite nice that you can see some localization, because Albert made a really great point about the importance of that earlier. Now,
Leonard, I'm sorry to interrupt you, but you ran out of time. So
yeah, I did, because I'm almost done anyway. On the 3D structure, you can see that the modifications are there. When it's dark blue, it means multiple sites. So you can see your proteins are really covered in modifications, like your dinosaurs are covered in feathers. And with that, I actually come to the end. And the end is that, okay, dinosaurs are not what we thought they were, but maybe that's not such a bad thing. And maybe there's this whole new generation of really cool things that we can find out about our dinosaurs, about our proteins. And if you're one of these youngsters in the field right now, I think it's a very interesting time, because there's lots of stuff to be found, there's lots of low-hanging fruit, and so the early adopters will probably get all of the worms here. So with that, I'm done. Thank you very much. And I hope that you have some questions for me, which you will chuck in the Q&A so we can answer them later. Thank you very much.
thank you, Leonard. And indeed, as Leonard mentioned, so all kind of questions, please write in the Q&A, and we will then ask them, pass them through the presenter. So Leonard, if you could stop sharing your screen. Thank you. Then we now move to Neil's talk. Neil is from the Northwestern University. He's going to give a keynote talk today. I'm very curious to see what that nuke-MS is all about. I think Leonard already gave a nice introduction to the field of how proteins look like, how these proteoforms look like when they are modified. I'm sure that Neil will deal with that in a bit more detail. So Neil, it's all yours.
Oh, Chris, thank you, and thank you to everybody to invite me in, and Tatiana and Albert and Leonard made some great points. Hello from Chicago, and you can hear me okay, Chris? Screen
Yes, we
okay?
do. Yeah.
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 Okay. Great. Yeah. Well, so which feathers are functional? That's one thing I'd like to pick up as a thought, and so let me jump right in and talk about Nuke MS and functional PTMs. There's a really clear linkage all the way back to 20 years or more about functional PTMs and combinations of them. So histones are a great place to talk about feathered dinosaurs or feathered nucleosomes. So let's look at how we can read out not just proteoform level information, so the combination of feathers that exist on your big animal, but also nucleoforms. So what about if you had several dinosaurs together in, say, a packaging of the DNA and via about 100 million of these nucleosomes that package our 1 meter long DNA into cells? And so I'll take you through that, this new method and data type called Nuke MS, as Chris mentioned, and we can talk about future directions going quickly into the Q&A. And so my comment about combinations of feathers is in this three-tiered worldview that I'm sure many of you have seen or thought about, that we can have mass spectrometric information be provided on one of these three tiers. And there's a lot of emphasis on bottom up, of course, and 3 or 4 billion euros a year industry that drives a lot of value generation in our field and assignment of function to proteins. So PTM detection and assignment of their functions is, of course, a major challenge, as Leonard so articulately noted. So that's why we, for years, have been focused on proteoforms and keeping things intact, where you gain 6 advantages that I won't drag you through, but one of them is stoichiometry, that you know, if you do top down, how much of the pool of proteoforms in a family that emerge from the same gene are in different forms, and what the PTMs, how they occur in combination, what their stoichiometry are, or occupancy, some people say. But let's go and take it to the next level. That's the native level, with the idea that the connectivity to underlying biology and the phenotype is tighter, of course, but the measurements are harder. And so as part of a consortium, this word proteoform tying to function at some high level has been successful to some degree. And this worldview is pervasive now in our field and even outside of the field of proteomics. So moving quickly into these 2 modes of top-down, if you keep your proteins intact, you're familiar with denaturing top-down, and there's a disadvantage, makes it hard, which is all these charge states that form. But if you go into native, you get far fewer charge states. So that means the signal in any given charge state is higher for the same concentration of sample. You also have all sorts of other advantages, like biologically relevant complexes stay intact. The linkage between native MS and structural biology has been established, hundreds of papers. Lot of great colleagues all around the world have been focused on this, and some on this call. And so let's do that for nucleosomes. It's really quite simple. And this reflects also a interest in the synthetic biology community, where they too are realizing that if you can synthesize whole nucleosomes and do assays, biochemical assays at that level, you will have privileged information. You will avoid artifacts through peptide level enzymology, where you can have promiscuity of those histone modifying enzymes and complexes at the peptide level, but you really suppress that when you feed enzymes whole nucleosomes or dinucleosomes. And that sort of parallels the evolution of analysis philosophies that I laid out. And so there's this synergy happening with those now able to synthesize nucleosomes with precise combinatorial control and create synthetic nucleoforms, as we now call them. And so we're going to match that synthesis ability, and you'll see that as I quickly move through the talk. But we're going to focus on the green native mode here and keep our nukes intact. That means we're going to have 200 kilodalton species, about half the weight is DNA and half is the octamer of 4 core histones, 2 copies each in the nucleosome core particle. And I'm channeling basically a paper, this paper on the right-hand side of your screen. And so we'll go through it in pretty short order. So here's a naked nucleosome, and I'll say nuke for short. But there it is. It's about 200 kilodaltons and native MS, not surprising. This has been done before, our work. And so we're simply just fragmenting or controllably disassembling the nucleosome, and when you do that with HCD, in this case, out pop the 4 histones and all their proteoforms. And so this is a naked one, and so it's quite easy to then do MS to the 3, and I'll come clean and say pseudo MS to the 3, and I'll make that clear in just a moment. But you can also get the fragmentation data for the proteoform and assert the position and combinations and modifications. And I'll go through that as well. So here's what I mean by pseudo MS3, if you haven't seen this before. This all was fired up in 2011 with a joint project with Thermo to extend the mass range in what became the UHMR. And because of the setup, native MS was pretty straightforward. In terms of extending the mass range of the quadrupole was a little more involved, but that allowed you to work at high mass, and that's also now a feature on the new Ascend or new Orbitrap from Thermo. And so what I mean by pseudo MS3 is that we can filter out a charge state and then eject in the HCD cell, and out would pop subunits of the complexes. And we've done this on endogenous complexes, et cetera, and published in 2018 on that for a native proteomics type experiment on hundreds of complexes from primary metabolism. But here's now the pseudo part. So when I fragment, I'm actually fragmenting in the source, liberating a monomer, and then fragmenting that monomer, but it wasn't in a true MS3 experiment. So I just want to make that clear as to not oversell what we're doing here. But if you do that kind of pseudo MS to the 3, and I hope you saw that animation okay, you can do that on all the 4 core histones and do pseudo MS3 and get bottom line proteoform information. So now let's start to get more complicated. Here we're treating a nucleosome with PCAF. That's a acetylating enzyme, a coactivator from a coactivator complex. And okay, so you treat it for an hour with acetyl-CoA, and the acetate gets transferred, and there you go. And yes, PCAF on a whole nucleosome should hit primarily H3 K14 acetyl. And the veracity of our evidence is borne out in these blue flags here, which map the B and Y ions. So we've got really nice localization here of K14 acetyl, and we know the stoichiometry. So let me make the stoichiometry point. Also, with treating now with PRC2, that's a complex, a co-repressor complex with the famous EZH2 enzyme. That's a methyltransferase, which is known to have the ability to do mono, di, and trimethyl reactions on H3K27. And so, and at these approximate rates, it matches what people know. The trimethyl's the hardest step for the enzyme, so it's the smallest amount. And the stoichiometry, these ratios are quantitative. And the dimethyl here localizes reasonably well to K27 dimethyl. So these are all in vitro, standard. We're just getting going. Okay, so now let's take HeLa cells. Let's go to endogenous nucleosomes. And so you're looking at the MS2 ejection experiment for endogenous nukes as a pool, isolated and created mononucleosomes from the nuclei of HeLa S3 cells. And so all of the 120 or so proteoforms here, we spent some time going through and doing the quasi-MS3 experiment, and did that for about 100 of these proteoforms above 1% relative abundance. And that allows you in a single spectrum to get a survey or a bird's eye perspective of the entire epigenetic landscape of those cells. It's notable that we don't have any localization like in ChIP-seq or other, cut and run type of experiments, if you know chromatin methodology. But we can get a real perspective and do A versus B comparisons and do it in a very facile way. That's a single spectrum snapshot of the epigenetic landscape of those cells. And I'll show you evidence of that or examples of that in just a moment. But, so what pops out is what you would expect. H2B has a bunch of members of a gene family differing by an amino acid or two. There's about 10 of those. There's about 15 H2As, and they are different functions for these isoforms or members of the gene family. But let's do what I consider my good old friend here, which is on 50% of all nukes, they have this friend as the H4 form, histone H4 proteoform. It is N-terminally acetylated and K20 dimethylated, and that's on 50% of all nukes in cancer cells. And that's just why I call it an old friend. And so it was easy to fragment and do the quasi-MS3 experiment. So let's get a little bit harder here, and maybe Chris throw up the 5 minutes when I get in danger of running out of time. Give me a high five. So we're going to now couple this nuke-MS readout with immunoprecipitation. And admittedly, it's a contrived system just to get us going, and that's a flag tag. So we transfected in a pretty crude experiment, but we transfected in a form of H3.3 that had a flag tag on it in 2 forms. And so I'm going to tell you the story of what co-precipitates out with that form of H3.3. Okay, and so we're going to do very standard things, create the mononukes, and do an immunoprecipitation, immunoenrichment for that flag tag. It's on, at least 30% of all the H3s are having the flag tag in these cells. After creation of mononucleosomes and flag tag, as I mentioned, IP, then we submit it to nuke-MS. Okay, so what do we see relative to bulk? Here's these MS2 level, or when all the histone proteoforms pop out of these nukes. And I'm showing you the HEC293 T cells in bulk here in the top. And in this butterfly plot, I've got this H3.3 immunoenrichment in the bottom. Okay, and if you know anything about H3.3, that particular gene of H3, there's 3.1 and 3.2 and centromeric H3s, but H3.3 is known to be associated with U-chromatin, or activated, more permissive chromatin. And so too was H2A.Z. So let me draw your attention down to this form down here. So we saw a 6-fold upregulation, six-fold increase in the abundance of H2A.Z, which is a specific variant of H2A, also known to be enriched in euchromatin and at the promoters of genes. And so there we get an immediate verification that we're able to now stitch together the composition of the nucleoforms that co-occur with this flag tagged H3.3. Interestingly, we can rule out massive isoform changes to H2B and H2A. They're just quite the same in these butterfly plots. But we also see evidence for eukaryotic marks or marks on histones H3 and H4 that are increased, that are associated with active transcription. And I'll show you one evidence of that in just a bit. But we verified all of this with good IP reagents for ChIP-seq. So these ChIP-seq runs are shown just example loci here in the bottom. And you can see H2A, perhaps H2A.Z. You can see that an AbCam antibody co-localizing with H3.3 here at the top. So as you can see, top to bottom, H2A.Z and H3.3 co-localizing. And there's the full roll-up genome-wide, and I'll spare us for time purposes. And a lot of these marks are known and have been done even in this cell line. Okay, so let's do one more thing here as we march toward the goal line and some Q&A. Definitely want to leave time for that. And that's to really ask what nucleoforms are present when you have this mutation. This K27 to M is a famous mutation about 10 years ago, discovered through genomics that leads to gliomas and pediatric glioblastoma. Well over 60–70% of kids who have this horrible cancer have this mutation in the H3.3 gene. And because it gets methylated, right, what other changes elsewhere on these nucleosomes form? And it's just this horrendous cancer, and I'll skip that part. But certainly at our children's hospital here in Chicago, we're very intent on finding this and even doing this type of assay out of cerebral spinal fluid for diagnostic purposes. Something we're still working on. So at least in cell lines, again, a contrived experiment, we transfect in now K27 to M, do the same immunoprecipitation, and compare the H3.3 wild type from the HEK cells to now HEK cells carrying K27 to M. And you can see that the flag tag is working. Here's the H3.3 part, which has the flag tag on it. Normal H3.3s appear in this spectral region. So here, one quick thing we could ascertain is that over 90% of the time, the H3.3s that occur in these nucleoforms, there's two copies, both of them have flag tags. And so that's one of the things that we could start to tease out with nuke-MS is the idea of heterotypic versus homotypic nucleosomes. That is to say, there's two copies of each histone. Are they symmetric or asymmetric in their modification profiles? And that's another thing that would be very difficult to tease out if you digest them all down to peptides. So I'm going to spare you a lot of the details, but we went through and compared the different forms here that occur in the K27 to M nucleosome. And that mutation is on the tail of H3.3. And so we were able to see upregulation of K16 acetyl on H4, and that's an activation mark as well. And so it was known that K27 to M is a poison of sorts, and it also creates aberrantly activational marks like K16 acetyl. And so we did ChIP-seq on this to verify, and it lines up with prior literature affirming the validity of the method, which is quantitative, by the way. So here, here's the K27 to M. You can see the K16. This is about 60% K16 acetyl, 40% K12, but that monoacetyl form is up in the K27 to M nucleoform. And these are the tech reps. These three little white dots here are the tech reps. So it's a quantitative method. K27 to M, of course, removes the methylation spots. So you can see it's the methylation equivalence is lower in the K27 to M because you're removing the methylation sites. And that, again, also affirms the method. That's logical. And again, H2A, there was this tiny change in this isoform of H2A, but most of them are not changed in the K27 to M. So let me just focus on just one observation that is a 15-fold. Yep. Great. High five, Chris. You got it. Yeah, so this is the sort of the key. If you're a mass spectrometrist, aficionado, proteomicist, let me linger on this a bit. So what is the primary data that allows us to assert the title on this slide, that we see an activational mark, so H3.3 K79 dimethyl. Normally, it's present at about 1% relative abundance. So, on the hairy edge of detection. But here we're seeing a 15-fold upregulation relative to that background in bulk chromatin. And here's the evidence that allows us to do this. So we did their nuke MS, right? And we're interrogating these signals by MS3. Okay, and it gets hairy, right? Because H3 is pretty gnarly. It's got a lot of different mods, a lot of methylation equivalents. It's the hardest challenge to nail down, especially as you get over 8 or 9 methyl equivalents. So what we've got here is this K27 to M. So we've got fragment ions like this one that would be consistent with that mass shift for the mutant. For homotypic nucleosomes, symmetrical for that mutation. And then these B and Y ions right here. So there's a B ion, there's a B ion, there's a Y ion. That allowed us to localize the methyl equivalents, and it's about half of a methyl equivalent is localized to that sequence region. And the difference between the B77, that's this one, and the B81, this one, that difference is plotted here. So the delta methyl equivalents allow us to assign, okay, we've got some methylation on K79, and we shouldn't, right? Relative to bulk, there would be 0 detectable or very nominally noise baseline detectable methylation, any methylation on K79. But here we have, and we then, how do we know if it's mono or di or tri? Well, it's mostly mono and di that we know. And then here's this ChIP-seq trace. This is a great antibody for this, and here's the ChIP-seq co-occurring with H3.3, and you can see here's the co-correlation square, this little cell here, and it's very high scoring, the co-occurrence of K79 dimethyl with H3.3. So, right, and it's not perfect. Of course, there's discordant areas, but it's pretty concordant in activation marks where this H3.3 lays down. A reviewer asked, "Hey, can you do mono ubiquitination? Can you do big PTMs?" And we were like, "Yeah, we could do that." Okay, so prove it. And so here's a synthetic nuke with H2A K119 ubiquitination. That we can detect by nuke MS. There it is, homotypic symmetrical synthetic nucleosome we got from that company, EpiCypher. As a last thing, readout, we're getting into capillary electrophoresis here. It's basically very small loading amounts, your 10 nanoliters, not 10 microliters loading amounts, and it can handle pretty complicated samples in terms of matrix, neutral, or salts. And so right out of the EpiCypher storage buffer, we could separate those nukes and get nuke MS data. And we have a paper on this. Here it is in passing. Kevin Yoos is a spectacular 4th-year postdoc and looking for jobs in Europe, by the way. That's it. So nuke MS, it's a thing, and they are tractable by native top-down MS. You can couple it with immunoprecipitation, and generally to line up with Leonard and faster readout is needed if we're going to detect and assign function to all these PTMs. So there's Kevin. I want to thank him in particular. He got great expertise in capillary zone electrophoresis. EpiCypher gave us the nucleosomes, and Luis also is notable. He was first author on the nuke MS and really charged after this. I want to thank Thermo for support and note my financial conflict of interest with Thermo. Also, Luis was fond of making artwork out of mass spectra. So he has a whole art book where he manipulated mass spectra like this one you can sort of see. And he's got all sorts of amazing artwork, and some of it's online. So, all right, Chris, I'll stop. Tatiana, thank you. Everybody at Epic, let's do some Q&A and get better together. So thank you.
Thank you so much, Neil. Very interesting talk. Thanks to all three presenters. Indeed, as Neil mentioned, let's go to the Q&A session. And I'm just scrolling down here. Let me see.
Maybe we can start. So there is one question from Albert to Lennart, who spotted, Albert spotted modifications on valine and noted that you had quite a few, and he's curious to know what it is.
Yeah, so typically these are N-terminal valines. So it's a modification on a valine that's on the N-terminus of a peptide. Now, the other thing that can happen, in fact, and this is a very common problem with an open modification search, of course, is that an open modification search does not see chemistry, it sees mass. And you can have equivalent masses. So, for instance, if you have a methylation on a valine, it could be very similar to the phosphorylations that you discussed on histidine, that you have a methylation on the arginine right next to it, for instance, and then the localization might be off. And that gives a valine modification. And so these kind of things probably are the biggest issue in all of these open modification approaches, because we fundamentally suffer in the mass spec from a lack of sequencing capability. So this is why we need search engines, right? Because some of the fragment ions are missing, and that can create these kind of funky things. And to the algorithm, it all looks very similar. And so there's various approaches that people are using, including ourselves, to try and figure that out, which is a localization score, but also minute changes in the retention time, especially on the N-terminus of peptides. That can potentially work because the N-terminal residues are more important for the retention time prediction. So yeah, there's definitely going to be some funky results in there, especially at this larger scale, where these kind of errors result in modifications that do not at first sight make a lot of sense. And so actually going through these things and splitting them into things that make a lot of sense and things that do not make a lot of sense, and then finding out in the latter category what caused them, is probably something that will keep our field occupied for a little while. But it's a necessary process, I think, that we have to get through in order to make this whole approach more mature and more high throughput in a way. Yeah, well spotted.
Okay, there was a question for Mariana. So somebody asked if you have evidence of crosstalk between arginine methylation and other protein modifications. And for instance, could the protein changes in the OOPS experiment when you use cisplatin also be due to other protein modifications?
Yeah, exactly. That's perfect the point, because we know from literature that there are frequently a crosstalk between arginine methylation and phosphorylation, for instance, on serine. So in the meantime, my colleagues are also trying to study the corresponding with the analysis and the enrichment with for the IMAC or titanium for phosphorylation after the hoops, so that we can investigate really if there are modulation that are in common and other ones that are specific for a modification for a PTMs and not for the other. Also, it will be interesting to see if proteins that are in some way upregulated or downregulated into the interface can have opposite results because of the different charge. Yes, of course.
Okay.
Okay, then one of the questions from Neil, from Chris, is did you ever consider genome editing techniques to endogenously introduce an affinity tag and mutation into histone, and then pull the nucleosome out of the nucleosome of interest out of cells?
Yeah, that's what did I call it? A contrived experiment, or a crude. Yeah, you're right. That would be much more precise to put it under control, the endogenous promoter, precision editing. definitely superior. We still have not done that, but that is superior. And we're going after immunoprecipitation of just endogenous forms, so no affinity tag, and making some progress there. So we're sort of bypass the affinity tag and going after what's endogenously there.
Okay. Leonard, I have a question for you. If you look at all the data that you gathered recently, is there actually any remaining human protein there that is not modified?
Very few. But then keep in mind that we also see a lot of artifacts as well. So we are not splitting biological modifications, biological artifacts, or chemical artifacts. As a result, practically any protein will be identified with something somewhere. And then, having said that, somebody asked me in another talk recently the question of, do you actually think that there are proteins out there for which not a single molecule of that protein will not be modified in a biological way? That's a very interesting question. It's probably something that we can have a very big debate about. I probably think it exists, right? There are proteins that are in so low abundance that it's probably plausible that this molecule never gets modified. But then again, who knows? The fact that we find phosphorylation everywhere, for instance, it might be that there's just no way that any protein can ever escape being phosphorylated. Whether or not that is a functional phosphorylation is a completely different matter, of course, and that's what Neil hinted at as well. But then an interesting question becomes, why does it get a phosphorylation? And what is functional? Does it have to be biology, or can it be some kind of other thing like sequestering of phosphoryl moieties? God knows what's going on. So the short answer is I do not know. I do not have all the evidence, but there seems to be a lot of modification going on, and a lot of biological artifacts as well, which I think are much more interesting than you would think at first sight.
Okay.
So I will ask a question which is written here anonymously to Marianna, which is, can you couple OOPS with specific enrichment of modified peptide? Is it feasible in terms of yield or not?
Yeah, we are trying, because sometimes this is really the problem. For instance, in the case of the RGE methylation, we would like to enrich specifically a methyl peptide after the hoops, but the commercially available antibody actually require a starting material at least 10 milligram. That's not so easy to obtain from the hoops. And we are in contact with the company. Probably we will be easier soon, because they develop an antibody that requires just a few milligram, 1 milligram or less. While for other type of modification, like in the context of, for instance, the phosphorylation, we know that it's require really low material, so it can be, it should be feasible, and we are trying.
Okay. I just see that there's a question. Somebody raised his hand. Juan, if you could ask your question, please.
Yeah, thanks. First of all, thanks for the talks. It has been amazing. So this is a question for Neil, because he surely mentioned something about occupancy or like abundance, or for example, of a specific proteoform. So I know that the special is more like a top-down, but what do you have to offer? What could the community should follow if they really want to measure occupancy accurately? And this is also linked to the fact, well, for that, you'll first be able to detect and identify any possible peptide with its specific peptidoforms. What is the best way that the community has to offer for measuring this if you don't get to the possibility of ionizing the full protein and assessing it that way?
Oh, yeah, I think when you digest and then go after peptidoforms, you make it very difficult to assert the occupancy or stoichiometry that peptidoform represents. So I'm pretty straightforward saying we have to go after the proteoform level information. It is not the case that peptidoform, even if you got 100% sequence coverage from all the peptides, it's not equal to proteoform level information. And that's, I think, partly intersecting with what Leonard was talking about. And I would simply love to be in Belgium right now, both because of the beer, but the company would be great. This topic is so vitally important for us as a community, because I don't think a lot of people have thought through all of the repercussions of what Leonard is talking about with biological artifacts, there's chemical artifacts, and then getting to the functional PTMs is, through stoichiometry, I view it as a filter. So if you say, okay, if you take Leonard's NF kappa B data or P53, you will find hundreds probably, or many, many dozens of PTMs. But when you look at it at the proteoform level, you will find less because it's a filter for stoichiometry and combinations. And so, Leonard, I just, as a last point, we just did this imaging, proteoform imaging technology, which is the most direct, low sample handling technique. And we see a lot of primary metabolism enzymes that exist unmodified. So a lot of glycolysis and TCA, admittedly, they're small proteins and they're super abundant, but the dominant proteoform is the one without any mods. So I'll stop there, Juan.
Okay.
Thank you very much.
Tiziana?
So there is a question to Lennart, who says, is anonymous again, who says, do you think that the problem of deep LC prediction with phosphorylated peptide applies also to other PTMs? And if so, which ones? As a matter of fact, just a second, see more, sorry. As a matter of fact, deep LC seems to work very well with oscillations, but what about all the other many more PTMs? How precise is deep LC globally?
Yeah, so these are good questions. Yeah. Before I answer that question, if I can just say something to Neil, because he made a very interesting point about the glycolytic enzymes. It turns out that in the top 50 most modified proteins, several glycolytic enzymes are there, and most of these modifications are biological artifacts, which is exactly what you expect in a way, right? Because they're constantly confronted with highly reactive intermediaries. That's what these proteins do. They make highly reactive intermediaries, and so I can imagine that they're not modified in the majority, but some of them do carry the modifications, and these artifacts are super interesting, actually, if you think about it. Anyway, now I'll get back to the question that was asked, because that's a great question. So the deep LC thing, there's another modification that was really poorly predicted if you looked at the slide that I showed, which was nitrosylation, because that again is also a very weird modification if you've never seen a nitrogen and oxygen combined. And so it's a generalization problem. It's exactly what you would expect. So the algorithm is a machine learning algorithm. So what it does is it finds patterns in the data that you give it for training, and then it tries to extrapolate from these patterns what happens in situations that are outside of the core training set. Now, if you've never ever seen anything with an NO on it, then maybe it's not so easy to figure out what an NO would do. And so are there other PTMs that have problems, and how do you in general expect this to happen? Well, if you have a PTM that has a very weird element in it that DeepLC has never seen before, I'm just making something up. So if you say I have a selenium-based modification and it has never seen a selenium anywhere, it will be very difficult for DeepLC to figure out what the effect would be of that modification, of course. However, since we do actually, of course, include phosphorylation and nitrosylation and so forth in the training of DeepLC, it can actually generalize pretty well, because biology does not play with that many elements when it comes to modifications. And as a result, the question on how precise is DeepLC globally, it is darn accurate. In fact, it's probably one of the best retention time predictors that are out there right now. It's competitive with everything that's out there, even the ones that don't do modifications. And some third-party evaluators have shown that DeepLC is really, really quite good. I think a very recent preprint from the MUN group showed that they use transfer learning to take something that looks very similar to DeepLC and make it slightly better. So there's a bit of a race on in trying to get it even better than DeepLC is. But I think right now we're probably limited, not so much in how accurate our algorithms are, but in how accurate our retention times can be established for peptides. Because that is a bit of a problem. We do small molecules as well, and small molecules have really nice retention times, but peptides do not. Peptides always have relatively broad peaks, especially compared to metabolites. And so as a result, I think we're really pushing the limits of what makes sense to make things better. Both for the unmodified peptides, where we have been for the last 2 or 3 years, and now DeepLC takes that to the modified peptides. So I think we have reached kind of peak precision there, unless somebody comes up with a better way of assessing the retention time of a peptide on an actual chromatogram. And as a complete aside, but still relevant, I think we are at the same stage for fragmentation predictions. So there are several fragmentation predictors, but functionally, when you actually use them in a piece of software like IonBot, there is no difference in any of them. It's very interesting if you look at the PROSIT paper from Matthias Wilhelm and Bernard Kuster in Nature Methods, that they actually compared to an older version of MS2Pip, and they roundly beat MS2Pip in the prediction accuracy there. But when they use both algorithms with Percolator, they get almost identical results. And the reason is that even though they have a better performance, this increase in performance is essentially negligible, because both of them are good enough to get Percolator to the next level. And I think that's where we are with these predictors right now. It's very exciting. They're as good as they probably will get. And that means that you can really start using them in earnest, which many people are. So yeah, it's cool.
I'm going to, I know that we're running a little bit out of time here, but I'm going to take the last question. And I see that Albert and also Per, you kind of more or less ask the same question, to my opinion. So Neil, first of all, you're more than welcome here in Belgium. We indeed know our beers pretty well, so take that as an invitation, open
and
invitation.
Chocolate, Neil, chocolate as well. Chocolate is important.
Yeah, yeah, I'm not into chocolate.
Do you have beer and chocolate at the same time?
Yeah, sure. But anyway, so the question is, can your work, can that be pushed towards single cell kind of analysis? So because Albert mentions that the number of nucleosomes that you have in single cells should be high enough. Yeah,
that's brilliant insight. It is, if you do back of the envelope, you can get there for either nuke-MS or denatured histone preforms at the single cell level. We aren't excelling at the kind of work that so many are focused on in terms of bottom up and pushing toward the single cell limit. So histones would be the perfect place to push on that. And we've dabbled. We've got to 100 cells in even with our what I'll call B level skills in that area. But yeah, I think you'll see in the next year or so that there will be advances right on that point, and histones are the natural starting point to look for.
Okay, thank you. I guess we need to end this workshop, but before doing this, first of all, take a look at this slide. But I really would like to thank all the speakers of today. These were very interesting, brilliant talks. So thank you very much for joining and for dialing in. That was really, I think we had a very nice workshop today. And I would like to invite you to the next workshop that was already announced by Tiziana in the beginning. So here it is. It will be held on Thursday, November the 3rd, also in the afternoon. It will be on plasma proteomics. You see the lineup of speakers there, and also the organizers, Albert, Philip, and Jochen. So the registration is open. Feel free to register to advertise this to your colleagues as well. I'm going to give the word to Tiziana.
Yes, thanks. I have nothing else to add. Just thank you very much to the speaker. The talks were different, extremely interesting, and I was also very happy about the Q&A with a lot of questions and a lot of discussion. So thank you all.